All right, welcome everybody. How are y'all doing today? Great. Happy second Tuesday of the month. I hope everyone had a fantastic weekend. Here, fantastic. Enjoying the rainy weather. Thanks for braving it out and coming to hang out with us here at Reef for our fishing friends. So a couple events we've got coming up. My name's David. If this is your all's first time here, I'll kind of walk us through some of the upcoming programs we've got at Reef, and then we'll go into our presentation. And how the presentation will go is kind of Drew will talk about whatever he's talking about, which is fish ecology.
Well, I'm making it up as I go.
Yeah.
So,
and after that, if you have questions, we can go through those as well. And we're looking forward to it. So upcoming, we've got our Ocean Invaders screening at the Florida Keys Brewing Company. It's going to be on Tuesday, April 18th at 7 p.m. It's going to last until about 9 o'clock. That is a PBS documentary done on kind of the lionfish invasion here, as well as the deep water lionfish traps that Reef has been involved in testing. We also have upcoming Reef Into the Blue Book Club on Thursday, April 20th at 8 p.m. And Katie is going to be leading that. So fantastic. We're looking forward to that. Also, we've got the Fish of Water 5K registration now open. Who signed up? I signed up. Oh, come on. That's not nearly enough hands. So everybody, if you haven't signed up yet, check out our website. You can get a really cool rash guard. All of the proceeds from that 5K go to our Oceans for All Fund, which allows us to deliver our educational programming for free to groups that need it. Also, very exciting for many of you, I am sure, Carlos and Allison Estape are returning to the Florida Keys in person. They will be here in person Friday, May 12th at 6 o'clock, and they're going to be updating us on kind of their adventures with the Smithsonian and take us through all the fish they've been seeing. And with that, does anybody have any community announcements? Anything coming up that we should know about in the Florida Keys?
Next Wednesday at the History of Diving Museum is our free monthly presentation, and it's on the crocodiles of South Florida.
All right.
7 o'clock, also on Zoom. So either in person at the museum or on Zoom.
All right, fantastic. Thanks, Bob. What's up?
Every third Wednesday, yes.
The Eco Discovery Center in Key West is opening this weekend.
All right.
Yeah, check it out.
All right. What's the Eco Discovery Center, Andrew?
It's the sanctuary's museum thing.
Sanctuary's open house. Very cool.
It's like a little. It's in the name, Eco Discovery Center.
Right up.
Place to go experience eco.
There's an aquarium there too, right? It's like a little mini aquarium. No.
No. No, not an aquarium.
Well,
maybe not anymore. It's been closed for last 2 years for renovations.
Oh.
So it's like all new, brand new exhibits and stuff. It's like a big official public grand opening.
Yeah, well, fantastic. Check it out. Me and Andrew actually checked it out when we were in Key West, and I was really impressed. If you get the chance, you're in Key West, it's completely free too, which is even better. One of the only things that's free in Key West. Okay. So other than that, we also have our Arts and Science Festival coming up Saturday, April 29th. From 10 a.m. to 3 p.m. It's going to be here at the Reef campus. It's going to be highlighting arts and sciences throughout the Florida Keys. And we're going to have booths, we're going to have displays, take-home bags for the kids, and we're also looking for volunteers for that. So if you have some time on that Saturday, you'd like to come help out, check people in, do some activities. At the end of Fish and Friends, see Madeline, or Moose, as you all know her. I think, is she in here at the moment? No. But if you see her afterwards, she'll be in here, and talk to her about volunteering, or email me, and we'll get it taken care of. All right. And last but not least, we've got our Florida Keys Lionfish Sweeps. We talked about this last time, but if you all are interested in getting a sweet free rash guard or an awesome custom lionfish buff, you can go out and spear lionfish with any of the local dive shops. And if you bring them here to Reef, you have one that's maybe too small for you to fillet, or maybe you're like, you know what, I don't feel like filleting today. You can donate that fish to Reef. We'll dissect it with our education programs here, as well as use the fins for the jewelry workshops, and the kids love it. There was actually one in here 2 hours ago. I don't know if you can smell it, but we cleaned up really well.
How about if you fillet it first?
Yeah, exactly. No, you've got to bring the whole fish. Whole fish, Bob. But there's also tons of cash prizes. Every time you bring a lionfish in, you're entered into a drawing. We're going to give away at least $2,000 in cash prizes. So definitely check that out. If you know anybody who's interested in that, who spears lionfish, who's not here tonight, let them know that this is going on.
Which dive shops?
You can go out spearing usually with Key Dives, Florida Keys Dive Center, and Islamorada Dive Center. Those are the ones I know consistently go out. I'm going to definitely check with the dive shop. Those are just the three I know. If you go down a little farther south, I'm Dive Isla Bella at a Marathon, Lou Key down a little farther, and then Captain's Corner in Key West. All right. And with that, I would love to introduce Drew Butkowski, who has been heavily involved in our lionfish program through the years. So thanks for coming, Drew. We're really excited to see you.
Thanks for having me. All right, it's my turn. Good afternoon, everybody, or good evening, I should say. Again, my name is Drew Butkowski. I'm a PhD candidate in the FIU Tropical Fish Ecology Lab. I'm really excited to have an opportunity to speak with you all today. Thank you for braving the weather, as David said, and thank you to all the wonderful staff at Reef for the great work that you do and for setting up this really cool event today. So I'm going to be talking to you a little bit about our lab's work here in the Florida Keys. You know, we do work sort of throughout Florida, but a lot of the really cool stuff we do happens right here in your own backyard. So it's exciting to get to talk to you about it. Before I go any further, the stuff I'll be presenting today, it's not just my own work, but it's a collaborative work of our lab, right? So it takes a village in science. So just to introduce a few people briefly, our lab is led by Dr. Alistair Harborne, who you see up here on the left. He's come to us from the UK. Those of you from an academic background, I don't need to tell you this, but behind every great PI or lead scientist, there's a postdoctoral researcher holding things together with PVC, glue, and duct tape. So ours is named Meg. Dr. Meg Malone came to us last year and has really been instrumental in some of the Fishscape work I'm going to be talking to you about later on. And then, as is typically the case, we have a bunch of nameless graduate students. The rest of us, there's too many to go through. I'll run out of time for my talk today. But rest assured, we study a ton of stuff from invasive species to parrotfish, feeding ecology, to reef fish, energetic expenditure. So our lab really kind of runs the gamut. So today I'm going to talk to you a little bit. We'll do a quick primer on coral reef ecology, really just to situate you to what are some of the background stuff that guides our lab's research, right? So what are the background phenomena that are guiding the questions we're asking, right? And that ties in again to some of the threats that coral reef ecosystems are facing today. Moving on from that, you know, after we kind of do our academic homework, we'll move into some of the research questions that our lab looks at, right? So we'll talk about how artificial reefs, right, or predators found on those artificial reefs might impact the behavior of fish on neighboring natural reefs. We'll talk about, you know, what makes good grouper real estate on coral reefs. Talk a little bit about Graysby habitat preference. And then we'll also talk about one of our newest projects, which is attempting to answer the question, if we want to protect reef fish populations, you know, how much seagrass do we need to support those populations? So quickly, a quick question. Who in this room enjoys fish ID? I think it's a pretty easy question in this room, right? Of course. So if you've paid attention, you may realize that folks in marine science, us fish ecologists, are fairly literal, right? So the yellowtail snapper, it's that snapper with, you guessed it, the yellow tail. So like I said earlier, our lab's called the Tropical Fish Ecology Lab. And with that, I'd like to conclude my presentation. Thank you for coming out. Pretty much sums it up. No, but to be serious, right, we do study tropical fish. The tropical part means that we don't like to wear wetsuits when we're in the water. The fish is self-explanatory. When I say ecology, I'm referring to the branch of biological sciences that focuses on the study of organisms, their environment, but really where the ecological rubber meets the road is how those organisms interact with their environment. As tropical fish ecologists, we get office views that look like this, all right? But I want to call your attention to more than just, you know, pretty office views. Within our tropical seascapes, there are a number of different habitat types. So these include, if you even look around the room here, but some of our principal components, our habitat types are our coral reefs that we know and love, coastal mangroves, right, which provide protection from storm, wave energy, as well as nursery habitats for a lot of important species. And then we have seagrass meadows as well. A lot of the work that our lab focuses on is the connectivity between reef and seagrass ecosystems, and I'll talk about that a lot more later. Now, why do coral reefs matter? I'm sure that plenty of people up here have talked to you about corals themselves. Corals are animals. They build the nice reefs we love. I'm a fish guy, so I'm going to stand up here and talk to you about corals as fish homes, as structure, right? So coral reefs provide the fish that we know and love. They provide them with refuge from predators. It provides habitat and niche space, right? It provides room for these fish to live and make a living. Coral reefs increase the food availability in the local area for fish, right? So it attracts larger predatory fish that are feeding on smaller coral reef fish. Reef complexity supplies nesting sites, right, for different damselfish or other species. And additionally, the structure of reefs provides protection from wave energy, right? So fish are able to shelter during rough storms and conserve energy that way. Now, it wouldn't be a science talk if I didn't introduce the villain of our story. If you've been paying any attention over the last couple decades, unfortunately, our tropical seascapes are in trouble. And we see headlines such as this: confronting the coral reef crisis, or a global crisis for seagrass ecosystems. Now, I don't have time to go into all of the reasons that these things are happening, but suffice it to say, we humans have a little bit to do with it. This is a time-lapse photo taken right here near Key Largo, right? This is Grecian rocks. If you've heard of it, I'm sure you all are familiar. And it's a sad state of affairs, moving from 1971 through 2004. Again, the loss of live coral cover is extremely alarming in the Caribbean and in the rest of the world. But I'm a fish guy, so we'll talk about it in terms of structure, fish habitat. In the 1970s, flat reefs made up only 20% of all Caribbean reefs when you look Caribbean basin-wide. As of 2009, that's up to 75% of all Caribbean reefs. And trust me, in 2023, that picture hasn't gotten any prettier. Conversely, in the 1970s, complex reefs made up 45% of all reefs. That number is now down to 2% as of 2009. Again, not improving anytime soon. The loss of reefs has the potential to double annual flooding damages and triple the costs associated with tropical storms. All right, so this is a really big problem that coastal communities here in Florida are well aware of, and we need to address moving forward. Now, what does this mean for fish? Right? I told you about all the reasons that we like coral, the structure, and what it provides for fish. Now, conversely, if we lose coral, live coral, and we lose the structure associated with those reefs, we're going to lose the important refuge sites, spawning and nesting and foraging habitats that these species rely on. So, admittedly, sometimes when I get up here and give these kind of talks, working in marine science can feel a little bit like this. But rest assured, there are people out here, in the Florida Keys National Marine Sanctuary, NOAA, there are people working on these very problems. So, as I said, we're not one to sit here and do nothing, wring our hands. One thing we've done in the Florida Keys, right, is the addition of artificial reefs. So there are over 300 active artificial reef permits in Florida. There are approximately 61 of those located within the boundaries of the Florida Keys National Marine Sanctuary. Now, these ships, these large ships, typically were old decommissioned naval or commercial vessels, which were sunk on purpose with the express purpose of increasing structure, fish habitat, as well as providing recreational opportunities for anglers and divers in the area. And they've done a pretty good job at those things. However, because we're fish ecologists, we always are concerned with the effects that human impacts can have on natural systems. And so we want to make sure when we're designing management plans or we're implementing an artificial reef deployment, that we're doing so in a responsible and thoughtful way, right? So we know that artificial reefs have higher densities of predators and lower densities of herbivores when compared with native reefs. We also know that artificial reefs have increased numbers of transient predators, which are more likely to move between habitats, right, and affect neighboring reefs. So in order to sort of study the impacts, we're using an artificial reef near and dear to the heart of FIU, right? So this is the FIU Aquarius Reef Base, which some of you down here may be familiar with, but it's actually located at about 60 feet below seawater off of Conch Reef, right on the elbow of Conch Wall. It was deployed there in 1993 in its current position. It bounced around a little bit before that. And it's unique in the world in that it's one of the only operational underwater research facilities in existence. Now, aside from all the really cool science that happens inside Aquarius, we're also concerned with the ecology happening outside of Aquarius because it's a large man-made structure that acts as an artificial reef. And the life support buoy above that structure supports a large number of barracuda, right? Much higher densities than you would find on a natural surrounding reef. Aside from barracuda, there are other predators that make this reef their home, right? So you can see the Goliath grouper that likes to hang out there. There are lionfish, graysby, and the like. So a quick primer on reef herbivory, right? So we're concerned about this large aggregation, what a word, of predators and how they might affect natural herbivory next door, right, on a natural reef. So normally on reefs, you see sort of like this. We have our parrotfish. They control algae, right, which are direct competitors of space with coral. If I were to add a large population of barracudas, now, number one, they may eat a few parrotfish. Those are called direct effects, right, consumptive effects of predation. But they may also just scare the heck out of all the barracuda, right, or all the parrotfish, excuse me. These parrotfish then may feed less, right? They have to avoid predation more, and that's something they have to balance with foraging. In this instance, you might see effects on coral recruitment, right? Because there's less parrotfish actively feeding, you may have an increase in algae, right? So this would be called sort of a fear effect. How do we measure parrotfish fear? All right, so we're interested in how are the barracuda on Aquarius Reef Base scaring the parrotfish on nearby Conch Reef? But how do we measure that? We can't send out a survey to all of the parrotfish. So we set out a series of grazing assays. So we used GoPro video stations, right, which you can see here. And we placed them at different distances away from the reef base, all right? The treatments in front of these, right, so we had seagrass. Then in front of that seagrass, we had a barracuda model, right? So it's designed to model a barracuda. We had a PVC tube as a control, so just a blank object that shouldn't appear threatening to a parrotfish. And then also just a blank seagrass assay, right? So no model in front of it. All told, over a number of seasons and a number of different deployments, we got over 450 hours of video. And this is sort of kind of what that setup looked like. On the left, you can see our barracuda model. All right. On the lower left here, you can see a seagrass buffet with a red band parrotfish helping himself. The little barracuda model in the background, you know, so they weren't that scared. And here we see again the GoPro facing our seagrass assay. Now, let's get to the cool stuff. So some preliminary results. These are the preliminary results of my lab mate, David Cohan's dissertation. He still has a few seasons left to run, so please do take all this with a grain of salt, but he's graciously allowed me to share it with you all today. We see a cool pattern. If you look up till about 0 to 350 meters, you see the two lines, right, representing no barracuda model and barracuda model between the blue and the red. You see that they overlap to a fair degree, all right? And the error bars between them almost completely overlap. So we can say that there's really no difference in either there being a barracuda model or there not being a barracuda model within 350 meters.
What's being eaten? Percent eaten, what's eaten?
That would be that seagrass assay right there. So this is the...
So the parrotfish are going after seagrass?
That's correct.
Not algae.
They're eating leafy greens down there. Yeah,
Okay.
so algae, seagrasses, and the like. Thank you. For those of you on Zoom, the question was, do parrotfish eat more than just algae? And the answer is yes, they do. So you see at this 350 meter break, we see all of a sudden that our barracuda model appears to get a lot scarier, right? And this makes sense if you think of it in terms of acute versus chronic risk. And we'll talk about that in a second. Additionally, you know, leading on from this, we looked at not only the percentage of that seagrass that was consumed over the course of a video, we also looked at how many parrotfish did we see per video. And we saw a similar pattern where close to Aquarius, right, this sort of 0 meter site, we're seeing relatively similar numbers across all treatments, right? Similar numbers of parrotfish. However, there's a large effect of an acute risk of a barracuda model being present farther away, where you can see that this barracuda model suppressed the number of parrotfish we saw. The way that we've explained this thus far, and it still needs to be borne out through statistical analyses, but we see this as being representative of a chronic level of background risk around the reef, right, around Aquarius, that the parrotfish have essentially had to adjust to, all right? So because if you were a parrotfish living within that 350-meter buffer, you can hardly afford to stop eating every time you see a barracuda because you're likely to almost always see a barracuda. But farther away from the reef, we see an increased effect of acute risk. So you can afford more to take a break from feeding when you're farther away from the reef. And so essentially this means that, at least in principle, our native fish may be balancing that background level of chronic risk with their foraging opportunities to make strategies that still allow them to function normally, which is good news for artificial reefs. All right, so moving on to a different thread, we're going to talk about what makes prime grouper real estate on reefs. Sorry, was there a question? Anyway?
It's just a thought.
Okay.
That, I don't know, this may be crazy, but maybe when they get close enough, they recognize they're not barracuda.
It's certainly possible, and I'd love to talk about it afterwards if we could. So we're afterwards. I will just ask that we hold questions till the end, just so I can get through this. Thank you. All right. So we're going to talk about Graysby. All right. They may not be the first fish that springs to mind when we say grouper. However, they are grouper. They're very important mesopredatory fish on our reef, and they're important. So who remembers why reef complexity is important to fish? This is your quiz. I'm just kidding. So it's important as refuges, spawning sites, and foraging and providing foraging opportunities. All right. So remember, complexity is good. Now, traditionally on coral reefs, ecologists were limited to measuring rugosity or complexity in 2 dimensions. It's called rugosity. And so what you would do is over, you would have a 1 meter chain. You would lay that meter of chain out over a reef, and then you would take the ratio of how long that chain went to 1 linear meter, right? So it gives you a 2-dimensional sense of how rough or rugose your reef is. In 2023, we can do a heck of a lot better. So I'm going to talk to you guys a little bit about 3-dimensional reef modeling, all right, and what we do in relation to that. So this is a process that's called structure from motion or photogrammetry, and it uses specialized software to stitch together individual photographs that you've taken of an area into an orthomosaic, right, or a digital, composite that you can see here on the right of one of our study reefs. From these orthomosaics, you're also able to create a digital elevation model, right? So you can get into the minutia of the cracks and crannies of the reef. And that allows us to calculate complex metrics of complexity beyond just rugosity at a given place. And it also allows us to take those complex metrics and expand them over a patch reef scale rather than just single point measurements of rugosity over a reef. So we're going to use these complex, or these improved, I shouldn't say complex, but these improved metrics of complexity to increase our understanding of why and how and when Graysby are using specific areas on the reef. To the left, you can see a representation of a digital elevation model where you have higher elevations in light, cool colors, and the darker colors represent lower elevations. And on the right, you see the corresponding orthomosaic, again, just a stitch together from a series of GoPro photos, transects on a reef, right? But we're able to do these really cool 3-dimensional reconstructions. We're able to calculate things such as surface area to planar ratio for each square meter grid cell. Also able to calculate maximum height, the slope of the reef, and also a really cool metric I want to talk to you a little bit about is field of view. So field of view has sort of been overlooked when it comes to reef fish, partially because of software limitations. So we now have software that will allow us to calculate view shed on reefs. What that means is, when we're trying to figure out why does a fish use a given reef cell, why are you there? What has brought you to that place? View shed has a lot to do with it. If I'm a visual predator, right, I want to perch in a place that allows me a field of view with a lot of prey in it, right? Or maybe I want to be hidden. Maybe I actually want to hide in a corner, right? You can think of different predatory strategies in your mind that might lend themselves to different areas on the reef, whether you're hiding behind something or you're in plain view. So this is a sort of a complicated, or busy, I should say, a busy model of all the work that we did. But we followed 16 individual Gravesby for 60 minutes each, right? So these were focal follows. You had a single snorkeler following a fish, writing down on a notepad where it is in a given space and time. We then also created these detailed three-dimensional constructions of these 6 different patch reefs. So we followed 16 fish over 6 different reefs. Those reefs were gridded into 1 meter square cells, and then we calculated, again, those complexity metrics I just talked to you about. This is what some of those outputs look like. So when you grid the reef into 1 square meter cell, you then get a resulting output, right? So these are a number of crevices per reef cell, with red being a very high number of crevices, a number of hidey holes. We see field of view here. Field of view is very limited in the center of our reef. If you look back, the center of our reef is the tallest part. And so we have a lot of tall things blocking our view here in the middle, all right? We also have maximum height of our reef cell. So you see the reef's tall in the middle, as we would expect. And then this is just time spent, right? So you as an observer, we're recording time spent. At the end of the day, we recorded again 16. We analyzed this in a regression framework. I won't bore you with the math, but trust me, Al did it. Just kidding. Maximum height was determined to be the key factor that explained the majority of the variance in Gravesby space use. All right, and you can see those sort of paired response plots here. So for both small and large grouper, we found that their maximum cell height of about 0.5 meter to a little over 0.5 meter to 1 meter for large grouper was a significant predictor of Gravesby using that cell. So again, that's a long roundabout way of saying, remember, structure is important. These Gravesby are preferentially using the tallest cells on our reefs, right? You can think of a number of explanations why that might be. It may offer them better foraging opportunities to ambush things below. Again, these are correlates we're looking at, but we do know that these are the habitats that they prefer, and that matters for future management plans. All right, and if you're really interested, you can read all about it here. Find me after the talk. I'd be happy to send you a PDF. All right. Now, next, we're moving on to our third and final sort of area of research, right? And that is to answer the question, the seemingly simple, but trust me, deceptively complicated question, how much seagrass do we need to support reef fish populations? Seemingly lots, right? But if I have to give that advice to a manager, they would like a little more specificity than just more is better. And so this work is couched within what is called the Seascape project. That's an acronym that I won't bore you with. But just know it's funded through the NOAA National Center for Coastal Ocean Science. So if any of those folks are listening, thank you. And it's a collaborative grant in FIU, right? So we're working across 5 different labs, right? And that's because we have to answer a lot of different research questions. So the first thing, we'll do a little bit more background information. I'd like to dispel your notion of the reef fish, all right? We use reef fish very generally. We describe them as the fish we see on our nice snorkeling trips or our scuba trips. However, many of those reef-associated fishes that we know and love are also commonly found in other habitats outside the reef, right? And you can see that illustrated on the right, where we have seagrass, mangrove, macroalgae, soft sediment. Please don't bore yourself reading all these numbers. The point is just to illustrate that many different families of reef fish use many different types of habitats over the course of their lives during both juvenile and adult phases. So if you're looking to create a coral reef fish management plan, it would behoove you to incorporate more than just coral reefs. Yeah, that's a GRE word for anybody studying. So Seascape Ecology, we talked about, we're ecologists by training. That means we look at the interaction between, organisms and their environment. And Seascape Ecology looks to answer ecological questions. How are organisms interacting with a heterogeneous environment, a variable environment? Remember, when we look out over the side of a boat, we see a vast expanse of water. But beneath those waves, there's a lot of heterogeneity. You have sand flats, you have seagrass habitats. Not all those seagrass habitats are the same. All right. So advancing technologies, particularly remote sensing, right, satellite imagery, has advanced our conceptions of how these systems function, right? How do coral reefs and seagrasses interact? All right. And a core theme of seascape ecology, and if you take away nothing from this portion of this slide, is only that the spatial arrangement of habitats matters. So if I have a coral reef, right, one coral reef is not equal to another coral reef because it also matters how much seagrass is around that reef. All right. You can have two identical reefs, but if they're in different contextual situations, you may find that they have different populations of fish found on them. So one of my favorite families of reef fish are the grunts, the Haemulidae, and they make these really cool trips between coral reefs and seagrass that I'm going to talk to you all about. So grunts are an abundant and ecologically important family of reef-associated fishes found here in the Florida Keys. Right? Much of what we know about them comes from the blue stripe, the French, and the white grunt that you see here. Now their diet, they're primarily benthic invertivores. That means they're feeding on the bottom, the benthos, all right, and they're eating small shrimp, crabs, and other crustaceans and invertebrates that they find there. What makes them really interesting for us is that they make these daily movements away from and back to their home reefs. So they aggregate during the day around these reefs with high structure. Remember, complexity is important. And then individuals disperse at night to forage in surrounding seagrass or sand flats. They then return to those home reefs at dawn, and the cycle continues. So they provide a really cool model system for answering questions about, how much seagrass do we need if we study their movements. So currently, as many of you in the room are no doubt aware, the Florida Keys National Marine Sanctuary gives special protections to these important hard bottom habitats, structures important, that are located within sanctuary preservation areas or SPAs. All right. However, managers also want to know now how much seagrass habitat should be required to support fish populations within these SPAs. So we've done a great job at protecting these hard bottom habitats. You know where they are, right? But they don't function in isolation. Those fish are moving outside of those boundaries, and they also are using habitat that should be protected. So to answer this type of question, our group is collecting data to find out, how prey availability varies across different seagrass habitats. So there are continuous seagrass beds, discontinuous seagrass beds. How is that prey varying? Which seagrass beds matter more? We also want to know how much do different reef fish species use different habitat types. That'll help inform us. How much habitat are they possibly using, right? If you protect more than they ever go to in their entire life, it's not necessarily adding any value. Not that habitat shouldn't be protected, but again, we're looking for sort of optimal solutions here, right? Where everybody can use resources. And then again, we also want to know, how does fish energy expenditure vary across habitat types? Because that informs estimates of how much seagrass. It feeds into bioenergetic models, again, which are used to calculate this. All right, so the first question I mentioned was, how does seagrass vary in its invertebrate abundance? And to do that, we get to do some pretty fun dives that we call foraging habitat assessments. You can see me here on the right. I even picked this picture where I was wearing my reef, my reef rash guard, so I'm on brand. And so we essentially do a 30-meter long net sweep along the bottom. It's a 1-meter wide net. It's bounced along the bottom, and that collects any small shrimp, crabs, and again, potential grunt prey items in the area. We then fold them up, take them back to the lab, and weigh and quantify what was there. We're also quantifying the physical characteristics of seagrass in that area. And all of this combined is used to generate a predictive model of prey abundance in the Florida Keys. So already we've found that seagrass habitats closer to shore tend to have more invertebrates, so higher biomass, as well as denser seagrass, which you would sort of expect. All right, next up to the fun bit. We get to talk about tracking fish. So this is the fun part of my work, and I'm happy to share it with you all. We are tracking fish throughout the Florida Keys using acoustic telemetry, all right? And we're tracking 4 different fish species. Acoustic telemetry involves catching and releasing fish with a small acoustic transmitter inserted into their body cavity. So we catch these fish on hook and line, take them on board. They're kept in a cooler with fresh seawater and just a little bit of an anesthetic. And then we do a small surgery, insert an acoustic transmitter, sew them back up. We make sure that they're ready to swim away. Here you can see one recuperating in fresh seawater before we ultimately release them. Those fish then swim around our seascape happily, hopefully, and we track their movements using a passive array of acoustic listening stations. So I haven't put every single receiver on this map. It would become far too crowded for your eye, but this is the seascape here off Isla Morada that we're working on. This is actually Tavernier Key to orient you, and our work actually runs from about Tav Key up to Rodriguez Key out to Pickles Reef, you can see right here, right? So you have acoustic receivers all the way out there. There's 31 receivers in this array. We also have a fine scale array where we're getting sort of positional tracks. That's 16 acoustic receivers, and this, sort of bounds a patch reef here in this middle box. So we can zoom in here, and that's where this is. In practice, these are what these acoustic listening stations look like. They're stationary in place. We know their GPS point. So when a listening station detects a tagged fish passing nearby, it records both the time and a fish ID. We also have a current meter located out there. These current meters are kind of cool. In a straight upward position, they record zero current, but it has a magnetometer. So when the current blows it over, it's able to tell you both speed and direction. Somebody smarter than I did came up with that one.
Is this all real time, or do you have to go collect the data?
So the question is this data collected real time, or do we have to go get it ourselves? That's an excellent question. These are all data loggers. So we have to physically go out and download those data. So they're not live streamed. Overall, we've tagged 50 individual fish right here off the Florida Keys, and we'll repeat this project actually 3 times in the middle and lower Keys later. So this is going to be in triplicate. We've tagged 16 white grunt in 2 phases. We've tagged 15 yellowtail snapper in 2 phases. And then because they're harder to find and harder to catch, mutton snapper and barracuda were sort of caught over a longer period of time between July and October. So we're able to, again, track the habitat use of these fish. We're able to see what seagrass patches they use the most. We're able to see where they're going. We also want to see how much energy are they using. So I wore mine as a prop, but if anybody in here has an Apple Watch on or you've used a Fitbit before, you'll be familiar with this technology. The tags that we're using have an accelerometer. So we're able to track in 3 dimensions the acceleration of our fish over time. We're then able to, one of our master students is working to calibrate that acceleration with oxygen consumption, actually energy expenditure. And so we get graphs that output graphs that look like this. And this is the activity, right? So acceleration in meters per second of a white grunt over a 24-hour period. All right, if you look at the x-axis, it's actually a white grunt Christmas. So you see that this is sort of a 24-hour period in our activity. This is universal coordinated time, UTC. So this line here actually represents 7 a.m. local time, which is about sunrise. If you'll remember, our grunts are getting back to their home reefs right around sunrise, as you see a decrease in their activity levels, which is really interesting, right? So we're able to see with these tags that our grunts are performing as we expect them to. Please do stay tuned for more Fishscape updates. We just got our first summer of fieldwork done, so a lot of the data is actually still coming in. Again, as your question suggests, we're just really getting those downloads in the door. So do please stay tuned. We're doing a lot more cool work down here. Really quickly, wrapping up, just because my talk was titled Planning for the Future, our lab also does a lot of cool work in modeling coral reef fish populations around Florida. And so this is just quickly, if you're interested in this sort of work, one of our former postdocs, Rachel Zerker, put out a really cool paper modeling, coral reef fish biomass in Florida's coral reefs. And not only did she model current biomass, but she modeled predicted change of biomass in response to different management scenarios that are being implemented in the Keys right now. So we have the addition of the Mission Iconic Reefs, both phase 1A and phase 2, where they go from 10 to 25% coral cover. But, and I'll leave you with this note, the biggest thing that came out of all of her models across the Florida Keys Reef Tract, as in terms of both a predictor of fish biomass and a potential improvement, right, for the future, for improving biomass, is the addition of structure. So things like reef balls, artificial reefs, at least from a fish-centric perspective, stand a good chance of making a difference. So thank you all. I'd like to thank all of our funders. Again, science takes a village, and villages are expensive. So FIU, the Tropical Fish Ecology Lab, and then again, all of the wonderful agencies you see listed below. So I'll wrap that up. If you have any questions for me, I'm happy to take them in person. If we run out of time for whatever reason, feel free to check us out. We have a website, tropicalfishecologylab.com. And then this is my boss's email, not mine. Thank you all. Yes, sir.
How fragile is the seagrass to grounding? When boats ground on it, how long does it take to recover? Is it really stressful for it?
Okay, thank you. So the question is, how long after, say, a boat grounding does seagrass take to recover, right? Is it damaged by that? Now, I'll answer with the caveat that I'm not a seagrass ecologist. I study seagrass as fish foraging habitats, but I do know that when you damage the roots and the rhizomes, right, of those seagrasses, they can take a very long time to come back. I would imagine that the damage from a grounding is similar to the damage from a prop scar, right, of somebody running a flat. And I know that those can take on the order of years to recover fully. So unfortunately, they can be delicate ecosystems. The biggest problem facing seagrasses in Florida, though, I will say, on a broad scale, is water quality. So, you know, if you're a Floridian, you know. Any other questions? Yep.
Yeah, you didn't specifically address whether the seagrasses proximity to the coral reef is adjacent to the coral reef or say in the bay. So it matters, I'm sure. And we have a lot of protections in the bay as well. And when you're swimming around a coral reef, you often see seagrass around the coral reef. So could you talk a little bit about the distance? I mean, the fish migrate, but they don't go from the bay to the coral reef every night.
Sure.
That's more of a life cycle.
Okay, so, and correct me if I paraphrase this incorrectly, but I think the question was for the folks on Zoom, if I could talk about the influence of proximity of seagrasses to coral reef fish, right? And I apologize if I wasn't clear about this, but you're absolutely right. So seagrass that's outside of reef fish's home range doesn't matter from the perspective of that individual fish, which is one of the reasons why we're doing the acoustic telemetry to really actually track the home ranges of these fish. So we'll have an idea from our acoustic telemetry of how far away from reefs these fish are ranging. So you know what seagrasses are, in that case, ecologically relevant. So I hope that answers your question.
So this talk tended to, I guess the scale you were talking about is in hundreds of meters, not thousands.
It would be on the order for a grunt. It would be anywhere from a couple 100 meters to, say, a kilometer. For a barracuda, that could be anywhere from 1 to 10 to 20 kilometers. You know, so we're working across species, and I should have mentioned that more. For grunts, again, they're using very small areas, a couple 100 meters at a time, a day, per day. So these are individual foraging migrations away from and back to a reef. And those are on the order of about 400 to 500 meters.
I have a question about the more nerdy question about the passive.
Hit me with it.
The passive acoustic trackers.
Mm-hmm.
How do they work?
Sure. So the question was if I could explain a little more how passive acoustic telemetry works, and I will. I'll go back a few slides for you. So this was in my effort to fit a lot of lab research into a given time slot. I could talk for a while. So This is an acoustic receiver. Now, up at the top of it, this is the business end. This is a hydrophone. So we insert an acoustic tag. This is a USB, but for our purposes, it'll be an acoustic tag. And it has, this has a battery, right? And it pings an acoustic signal at, say, 69 kilohertz at a given coded interval. Say, for ours, it's roughly every minute, right? Our tag is yelling, essentially. You can think of acoustic telemetry as a game of Marco Polo, truly, right? Where you say Marco Polo, Marco Polo. Now, this is screaming Marco constantly, every minute. And we have a series of passive acoustic listening stations. And every time they hear Marco, they write down Polo. Right? Make sense? So that's how acoustic telemetry works. And so a lot of,
Passive is what threw me. So what's
Yeah.
passive is the listening station.
we have passive acoustic listening
I
stations.
was wondering how a passive, something in the fish would have.
So the reason we differentiate passive versus active acoustics actually refers to a field of marine biology where you're using like sonar to track fish schools. So you're actually pinging sound at the fish as opposed to listening to it. Sure.
How long is the two part, how long is the battery life, and is the surgery, they don't come out of the fish after they're done, the battery just sits in there in the fish, and
Yeah.
how aggressive is it? Do you have fish loss when you do the surgery?
Sure. So sorry, could you repeat the first part of it? I got the second part. Go ahead.
How battery life?
Sure. So the question is, how long is the battery life for these tags? And then the second is a little bit, let's talk about the acoustic telemetry surgery, right? All right. So we can go back. Oh, no. There. So our batteries last, it depends on the tag. So our smaller fish have shorter battery life, right? Because we're using smaller tags. Our larger fish, the barracudas, their tags are about 3 years, 3 to 5 years. However, our grunt tags are on the order of 90 days. So there's a large variability there. We're able to track larger species for longer because their tags are able to hold more battery. It's just simple as that. Now, as far as tagging surgeries go, we like to think we're pretty good. So we practice this a lot. Of course, as with anything, we have some mortality events, right? So you can tell when a fish has been eaten. However, it's pretty unlikely that that's actually the direct result of our surgery. We do a lot of snorkel releases, right? So where we actually individually take a fish and put it in a hole. And then also anecdotally, we had a pretty cool, this is a pretty unique scenario where we actually recaught one of our tagged fish 6 months later, or excuse me, 3 months later. It would have been at our next tagging event. So we caught one of our tagged fish 3 months later. He really liked that fish trap and I guess had gotten away with it the first time. And the sutures were completely healed over. We didn't actually know for the longest time that we had recaught him until we went to do another surgery. That's another story, but.
But isn't that there a tag sticking out like a?
So for the larger individuals, we do use an external marker tag, but for the smaller individuals, we found that the streamer tags are a bit of a dinner bell, and they get eaten more that way. So we try to keep them as natural as possible.
What happens when a predator eats a fish that's got a tag?
So the question is, what happens when a predator eats a fish that has a tag? So, or when a human catches it. Now, I can speak definitely to the human aspect of it. We have a really great engaged community here in the Florida Keys that's interested and passionate about marine science. So we've actually had 2 barracuda captured and our tags returned to us. So we have a streamer tag with a please call if caught number. And we've had great success with that so far. So, our first choice would always be that our tagged fish stays in the water. But we can't be the choosers of that, right? We caught them once. Evidently, they're prone to capture. And so we've gotten tags back from fishermen that way, which, again, we can't thank people enough for. It's a really huge help. And then as far as a predatory fish goes, that is something of an art form. right? So the tag itself will continue to ping continuously, assuming that it hasn't been damaged, right? So so it's robust to stomach acids. It's actually, you know, it's been embedded in a fish's stomach. It's designed to be inert, so the tag shouldn't digest. What will end up happening more likely than not is when analyzing your fish's behavior, you'll get a wildly different movement signature for about the gut transit time of your predator, is what I'll say, and then it stops moving. Right. So eventually, you know, a shark or a larger grouper or a barracuda will pass that tag because, again, it entered through the gastrointestinal tract, not the body cavity like we've inserted it. Cool.
Not so much of a question, but more of a comment, kind of piggybacking off that.
Everyone's
I know
favorite.
Anovacy does make tags, acoustic tags, that when it is eaten by another fish, when the stomach contents start to digest, it actually changes the number of the tag.
Yep. So Novasi has a tag that when it detects stomach acid, there's a pH sensor, and the tag ID switches from odd to even. So those exist. However, they don't have the accelerometer packages that we're interested in. So those are kind of a newer technology that they're working out the kinks for. Anybody else? Well, thank you so much. Truly, the pleasure's been all mine. Thank you.
All right, Drew, thank you so much for coming. We really appreciate it.
Thanks. You shouldn't have.
Drew has been around Reef for quite a while. He actually was Dr.
A picture?
He was a master student of Dr. Ali Candelmos.
No masters.
No masters?
I was just staff, research.
You were just staff.
Yeah.
I did not know that. That is something I've been telling people for far too long. Wow. So good to know. Drew was Ali's staff. Well, guys, thank you all so much for coming. Next month, on the second Tuesday of the month, we'll have Catherine Uden from Oceana talking about plastic pollution. And then that same week on the Friday, Carlos and Allison will be talking about their adventures throughout the Caribbean and through the tropical Eastern Pacific. So thank you all so much for coming, and we look forward to seeing you soon.
Thank you all.
I've been